Today we're going to talk about the circulatory system, and we have to talk about this starting with the blood. Now, the purpose of the blood is manyfold, but there are three major purposes of blood. The first is to transport. The blood is necessary for transporting oxygen and nutrients to the tissues, and also for transporting waste products and carbon dioxide from the tissues back to the lungs. Secondly, the blood is a regulatory tissue. It regulates temperature, regulates many other functions within the body. And then thirdly, it's protective. The blood contains white blood cells and antibodies that are important in the destruction of invading organisms, as well as cleaning up and protecting us from dead tissue that might occur during the process of metabolism. Now, when we consider the composition of the blood, one concept that's extremely important is the concept of viscosity. First of all, the blood has to be able to stop coming out of an injury once an injury has occurred. And hemostasis is that process that stops bleeding once an injury occurs to one of the blood vessels. Viscosity is the resistance to flow and the alteration of the shape of the flow due to cohesion. Now, the term cohesion means molecules of the same type sticking together or sticking to each other, as opposed to adhesion, which is molecules of different types sticking to each other. What you can see in the diagram here is two representative viscosities. The one on the left would be said to have a lower viscosity than the one on the right, and you can see that the one on the left not only comes out more quickly, but also forms a different shape when coming out and falling, and forms drops and a smaller stream compared to the one on the right. And this is due to the viscosity of the liquid. Most people, when they talk about viscosity, they think of the term thickness. Now, the combination of the red blood cells, or the cellular elements in the blood, and the water in the blood is what makes up the viscosity. And the percentage of cellular elements is a maximization of both the viscosity and the carrying capacity of the blood. Now, there are other factors in the blood that are under consideration. The density of the blood, which is the mass per unit volume. The acid content, which is also called the pH of the blood. These factors are also tightly controlled because they're important in the chemical reactions that occur within the body. Now, when we think in terms of composition, the blood is essentially a little over half plasma and the rest formed elements. The plasma is the liquid portion, and it contains essentially water and proteins. The formed elements are the cells that make up the blood, and they include red blood cells, white blood cells, and platelets. And as you can see here, about 55% of the blood is plasma. About 45% is the formed elements. Of that 45%, about 90% of that is red blood cells, and the rest is white blood cells and platelets. Now, when we look at the composition of the plasma, as I've said, it's mostly water, but it's also made up of some proteins. The most common or abundant protein is albumin. And albumin is important as it acts as an osmotic particle. Now, we've talked about osmosis before, but osmosis is the ability of a material to hold fluid within the blood vessels. Blood vessels are semi-permeable membranes, and they hold water. The water is held in the blood vessel by the presence of the albumin, one of the things that holds it in there, in the blood vessel. The second protein that's found is the antibodies. Now, antibodies are proteins that fight infection. They're produced by inflammatory cells, and they're there to fight infection. Other functions of globulin proteins are to bind proteins that are in the blood. And then lastly, the third protein that's found in high concentrations within the blood is fibrinogen. And fibrinogen is an important protein in blood clotting, and we'll talk about this in just a little bit. When we look at the various formed elements in the blood, you can basically divide these into three groups. The first of these groups are the erythrocytes, or red blood cells, whose primary function is to carry oxygen. And oxygen is carried within the erythrocytes in the form of hemoglobin. Oxygen has a very low solubility in blood. That is, you cannot dissolve very much oxygen physically in blood. Therefore, the blood has to have a protein that'll carry the oxygen and bind the oxygen and carry it. And that protein is hemoglobin. If you didn't have hemoglobin in the blood, then its ability to carry oxygen would be very limited. The white blood cells, on the other hand, and you can see that there are several different kinds of white blood cells as pictured in this table. They perform various defensive functions, and their defensive function is different depending on which type of white blood cell that they are. For example, neutrophils are primarily responsible for attacking bacteria, whereas lymphocytes and monocytes are more into attacking viruses and fungus and various other types of organisms. The eosinophil and the basophil has a more specific function that won't be discussed at this time. Platelets are actually fragments of cells called megakaryocytes, and these fragments float around and they become bound to injuries, and they help form the clotting function that occurs when the blood has to be stopped at an injury. This is a diagram of hemoglobin. Hemoglobin is a very complex protein that's made up of 4 subunits, 2 alpha subunits and 2 beta subunits. Each subunit has a binding site called a heme group. The heme group binding site has iron in it, and each binding site can bind 1 oxygen molecule. So in a hemoglobin molecule, you have 4 binding sites, so you can bind 4 oxygen molecules. Now, there's a lot of hemoglobin in each red blood cell, and so there's a lot of binding that can occur. Hemoglobin, as I said, is the protein responsible for carrying the oxygen in the red blood cell. Now, when you do not have enough hemoglobin, or for any reason there's an inability to carry enough oxygen to meet peripheral needs, that's what's meant by anemia. Some people define anemia as a decrease in red blood cells, but that's not actually correct. The correct definition is a lack of sufficient oxygen carrying capacity to meet needs. And you should remember that definition for anemia. Now, when you look at how white blood cells work, there's various things that have to be taken into account. First of all, is the ability for a white blood cell to pass through the blood vessel and reach the tissue spaces. And you can see on the right of this picture the process of diapedesis, which is the white blood cell passing through these small pores in the blood vessel and going into the tissue to engulf the bacteria. That engulfing is attracted by a release of a chemical substance that actually attracts the neutrophils to this site. And that process of attraction of cells by chemical stimuli is called chemotaxis. Now, once the white blood cell encounters its target, it eats it. And essentially, engulfs the foreign material or the the bacteria. And that process of engulfing these bacteria is called phagocytosis, and you've heard this term as well. We talk about pus. The term pus, some people find kind of gross. Pus is essentially the combination or mixture of dead and living white blood cells or leukocytes, along with foreign material that they've ingested. And so if you were to take a smear of pus, put it on a slide, and stain it, you would see live white blood cells, dead white blood cells. If the staining was proper, you might see organisms, bacteria, various other things that the white blood cells have eaten. And many of the white blood cells are dead because they ate something that essentially killed them. But in the process of doing that, the bacteria or whatever the foreign material is also destroyed. Now, we've mentioned hemopoiesis before because it occurs in the bone marrow. It's the process of producing white blood cells, red blood cells, and platelets. And this is a diagram of, probably more complex than you'll ever have to know, of how hemopoiesis starts. And it starts in the bone marrow with the multipotent stem cell. Multipotent stem cells mean that these cells can become red blood cells, platelets, white blood cells of various types. This differentiates or becomes more mature, and at that point it becomes either a myeloid stem cell or a lymphoid stem cell. At that point, if it becomes a lymphoid stem cell, it can only become a lymphocyte. If it's a myeloid stem cell, it can still become either a red blood cell, a platelet, or a white blood cell. Then they further differentiate, and once it goes into this next level, which is erythroblast, megakaryoblast, or myeloblast, then it's committed to one of those three cell lines. And ultimately it will become a red blood cell, a white blood cell, or a platelet. And you can see that erythroblasts eventually become erythrocytes, megakaryoblast eventually become megakaryocytes, which stay in the bone marrow and release little parts of their cytoplasm, and these are platelets or thrombocytes. And then the megakaryoblast, or the myeloblast, then becomes various types of white blood cells. And the monocyte is different than the other types of white blood cells, but still also different from the lymphocyte lineage, which is totally separate. Blood vessel becomes injured, there's a process of clotting which occurs, or hemostasis. Hemostasis is the process where the body stops blood loss. And this shows in very rapid succession the 3 steps of hemostasis. The first of these 3 steps is vasoconstriction, which is shown in the second from the top. First you have vessel injury. The second from the top is vasoconstriction, where the blood vessel makes itself smaller to decrease blood flow. The next step is the formation of a platelet plug. Certain chemicals are released at the site of the injury after vasoconstriction occurs, and it attracts these particle platelets, which bind together along with fibrin to form a plug. This plug acts as a building structure to put the coagulation part on top of it. The coagulation is the formation of various proteins that become interlaced and then build into the platelet plug and end up forming the full clot.